Assalamualaikum and hello everyone. Today we're going to present about polygenic and multifactorial disease, which is diabetes mellitus type 2. Without any delay, let's get started. What is diabetes?
Diabetes is a chronic metabolic disease characterized by hyperglycemia, which is elevated level of blood glucose that leads over time to a serious damage to the heart, blood vessel, eyes, kidney, and nerve. It can be characterized by diabetes mellitus type 1 and type 2. Today, we will look at diabetes mellitus type 2. Type 2 diabetes is characterized as a malfunction in the way the body regulates and utilizes glucose as a fuel. This long-term chronic metabolic condition causes an excess of sugar to circulate in the bloodstream. Generally, this disease can be described as insulin resistant due to the response to insulin being diminished. During this state, insulin is ineffective and is initially countered by an increase in insulin production to maintain glucose homeostasis. But over time, insulin production decreases, resulting in diabetes mellitus type 2. Diabetes mellitus type 2 is popularly known as adult onset diabetes, but actually, both type 1 and type 2 diabetes can begin during the childhood and adulthood, although type 2 diabetes is more common in adults rather than children. If not treated or taken care of, it might develop into other complications and leads to life-threatening disease. In this video, we will dive deeper to learn the insight into this particular disease, starting with pathophysiology.
Now that we already have a rough idea about diabetes mellitus type 2, I will bring you deeper into this topic by explaining about the pathophysiological aspect of this disease. But wait a minute, before we move on to the topic, let's review what a normal metabolism in healthy adults looks like. After every meal, it is normal for our blood glucose level to rise. And as a response to this high blood glucose level, pancreas will be stimulated to produce insulin to lower down this concentration. Inside the cell, glucose molecules will be broken down to form energy or can be converted into a form of storage and be stored. For example, glucose molecule will undergo glycolysis process to form energy in form of ATP. Now we have seen what a normal metabolism in healthy individuals looks like. But what about affected individuals? In affected individuals, this metabolism cannot be maintained properly due to the inability of the insulin molecule to properly bind to the receptors on target cells. This phenomenon is called insulin resistance. It can happen due to various reasons such as genetics, family predisposition, dietary habits, and obesity. As a result, glucose cannot be taken up by cells and accumulation of glucose molecules inside the blood circulation will happen. High blood glucose level inside the body will occur for a longer duration of time. Due to high levels of blood glucose concentration, pancreas will be stimulated to constantly produce and secrete more insulin. Persistent high blood glucose level will cause glycosuria, polyuria, and dehydration to happen. Glycosuria will then lead to osmotic diuresis and polyuria, which is loss of water and electrolytes from the body. This can be seen in form of frequent peeing in patients. Polyuria, on the other hand, will lead to dehydration in patients. And to overcome dehydration, our brain will be stimulated to increase our water consumption, which can lead to polydipsia. Prolonged dehydration can lead to decrease in blood flow flowing to the kidneys and can also cause renal failure to happen. Another common symptom of insulin resistance is obesity. Obesity is caused by polyphagia, which is the feeling of hunger and the urge to eat more. These are the pathophysiological aspects of diabetes type 2 in affected individuals. That's all from me. We'll move on to the next part.
Thank you, Balkis. Next, I will continue with the causative genes of diabetes mellitus type 2 or T2DM. As diabetes mellitus type 2 is a polygenic and multifactorial inheritance disease, its development is caused by several genes. However, in this video, I will only explain 3 examples of the causative genes that are commonly found. The first one is transcription factor 7 like 2 or TCF7L2. TCF7L2 is the most important type 2 diabetes gene with the odds of developing type 2 increasing by 30% to 50% for each inherited allele. The question is, how does this gene cause T2DM? The most likely primary cell target for TCF7L2 in T2DM is the pancreatic beta cells. When overexpression of this gene happens in pancreatic beta cells, it will disturb its function, which in return results in reduced insulin secretion. Reduced insulin secretion indirectly increase hepatic glucose output and lead to diabetes mellitus type 2. The next gene involved in this disease is ATP-binding cassette transporter subfamily C member 8 or ABCC8. ABCC8 is the gene that provides instruction for making the sulfonylurea receptor 1 protein, which is the important protein associated with the ATP-sensitive potassium KATP channel. The KATP channel is widely found in pancreatic beta cells and it controls the secretion of insulin out of beta cells into the bloodstream. When there is a mutation in this gene, it will disturb the opening and closing of this channel. The interruption that occurs will disturb the regulation of the insulin release, and this could lead to T to the M due to a blood sugar level imbalance. The last gene that I will explain in this video is solute carrier family 2 member 2 SLC2A2, or also known as GLUT2. According to a July 2013 study by RevSec, this protein facilitates bidirectional glucose transport. If mutation occur in this carrier protein, glucose cannot be transported across the cell membrane. This long-term condition will result in high blood sugar levels and diabetes mellitus type 2. That's all from me. Thank you.
Besides genetic causes, environmental risk factors known to impact the development of type 2 diabetes. For example, the environmental factors include unhealthy diet, obesity, and physically inactive. First, unhealthy diets occurs when people tend to consume high sugar and salt intake in their food. They like to eat highly processed items such as fast food, which tend to be low in nutrient, for example in vitamin, mineral, and antioxidant, but high on empty calories due to the content of refined flour, sugar, and sodium. And lastly, they like to consume food with more saturated fat. Secondly, obesity. Unhealthy diet associated with obesity. According to the studies, it states that type 2 diabetes rises with increases body weight. The prevalence of type 2 diabetes is 3 to 7 times higher in those who are affected by obesity than in normal weight adults, and is 20 times more likely in those with a body mass index BMI greater than 35 kg/m². Third, physically inactive. For example, no physical movement, such as lying on the bed for entire days. This sedentary lifestyle is one of the factors to develop diabetes and other chronic disease, for example, cardiovascular disease and hypertension. These environmental factors clearly play a major role in the development of diabetes, but they do not impact everyone in the same way. Even with the same environmental exposure, some people are more susceptible to develop diabetes than others, and this increased risk appear to be inherited. Hence, now we move to the next part, which is mode of inheritance. First, the time-plus risk of developing type 2 diabetes is 40% for individuals who have one parent with type 2 diabetes. Second, 70% if both parents are affected. The first degree relative of individuals with type 2 diabetes are about 3 times more likely to develop the disease than individuals without a positive family history of this disease. The concordance rate in monozygotic twins is about 70%, while concordance in dizygotic twins is about only 20 to 30% to develop the type 2 diabetes.
One of the molecular techniques used to detect diabetes mellitus type 2 is SNP's genotyping. SNP, pronounced as SNP, is a single nucleotide variation at a specific location in the genome that is responsible for the alteration of the protein structure and function, hence leading to the development of a disease or change in a response to a drug or environmental toxin. Thus, SNP has been used as a molecular marker to detect the early development of diabetes mellitus type 2 using primer extension nucleotide incorporation method, in which the researcher will design a primer and adding it to the 3' of the adjacent target SNP site. Then this primer will be extended using nucleotide polymerase enzyme. After that, the identity of the extended base will be determined either by fluorescence or mass to reveal the SNP's genotype by comparing the suspected SNP of a diabetic patient with the SNP of a normal person without diabetes mellitus. According to American Diabetes Association, there are 12 SNPs in 9 genes being shown in the slide that are normally associated with diabetes mellitus type 2. Moving on to the next method, which is exome sequencing. It is known as a next generation sequencing that is more time-saving compared to the conventional DNA sequencing. Exome sequencing is capable of identifying all of the vessels of an individual DNA protein region that provide instruction for making protein, aka exons that make up 1% of the whole genome of an individual. In diabetes mellitus type 2, mutation that occurs in the exon of the gene shown in the slides are thought to cause reduction in sensitivity of the peripheral tissue to insulin and reducing insulin secretion, leading to the development of diabetes mellitus type 2. In exome sequencing, the first step is to collect the blood from the diabetic patient, and then the researchers will extract the DNA fragment from the serum. This DNA of the diabetic patient will be captured using probe that is specific for capturing exome-only DNA fragment. Lastly, based on the exome DNA fragment that has been captured, it will be used for sequencing on a next-gen platform through bioinformatic analysis by mapping the individual exome-only DNA fragment reads of the diabetic patient with normal human reference genome.
Thank you, Insani. Next, I will explain about management for diabetes mellitus type 2 patient. Firstly, blood sugar monitoring. People with diabetes mellitus 2 are required to check and record their blood sugar level at least 4 times a day. The American Diabetes Association recommends to test the blood glucose level during these 3 times, which are before meals and snacks, before bed, and before driving or exercising. However, checking the blood glucose level is advisable whenever you think you have low blood sugar. Secondly is healthy eating and monitoring carbohydrate intake. It is important to consume more nutritious, low-fat and high-fiber foods such as fruits, vegetables, and whole grains. Registered dietitians normally will recommend eating fewer animal products and refined products such as white bread and sweets. Thirdly is physical activities. The initial step is to get your physician's green light to exercise, then choose activities that you enjoy. At least try to do 150 minutes of moderate aerobic exercise a week and no more than 2 days without any exercise. It is important to remember that physical activities can lower your blood sugar. Therefore, check the blood sugar level more often than usual. Next, there are a few activities of concern for people with diabetes mellitus type 2. First is being pregnant. The risk of complications during pregnancy is higher for people with type 2 diabetes. Thus, please seek and consult with your physician before you try to get pregnant. Secondly, life activities like driving and working can become challenges for people with type 2 diabetes. This is because low blood sugar can happen at any time, so it can pose a threat to the individual and also the people around him or her. For example, if your job scope involves operating heavy machinery or driving, sudden drop in blood glucose can be fatal to the person and surroundings. That's all from me. I will pass the presentation to Kairin.
Next, let's dive into the treatment of diabetes mellitus type 2. The first one is metformin. Metformin is generally the first medication prescribed for type 2 diabetes. It is a diabetic drug that is used in the form of oral medication. It works primarily by lowering glucose production in the liver and improving the body's sensitivity to insulin so that the body uses insulin more effectively. This means that metformin improves insulin resistance so that the insulin produced works better. It is available as a generic, so it is very affordable. Plus, metformin does not cause hypoglycemia when used alone. Metformin can also have other long-term health benefits. These include lowering cholesterol levels and reducing heart disease risk. Examples of metformin include Fortamet and Glumetza. Moving on, glucagon-like peptide 1 receptor agonists, also known as GLP-1RA, are injectable medications that mimic the effects of a substance the gut produces naturally in order to regulate appetite and blood sugar levels. Its functions is to make more insulin and reduce sugar release from the liver. These drugs also slow down the stomach emptying. When digestion is slowed down, nutrients in food are released more slowly. This prevents blood sugar from spiking after a meal and make people feel full longer. Examples of this medication include liraglutide, semaglutide, and dulaglutide. The last one is SGLT2 inhibitors. SGLT2 inhibitors affect the blood filtering function in kidneys by blocking the reabsorption of glucose into the bloodstream, thereby producing a reduction in blood glucose without stimulating insulin release. The unabsorbed glucose is then excreted in the urine. Examples include canagliflozin, dapagliflozin, and empagliflozin.